Okay, so we are at the top of the hour. Welcome and thank you for coming to the webinar series from the Ioneer Foundation called Sight and Sound Bites. This bi-weekly webinar series highlights research at the University of Pittsburgh School of Medicine. Today's topic, growing optic nerve regeneration. I'm Heather Cronus-Danek, Director of Development of the Ioneer Foundation. The Ioneer Foundation supports research to advance care for vision, hearing, balance, voice, and the cancers of the head and neck at the two world-renowned departments of ophthalmology and otolaryngology at the University of Pittsburgh. The funds we provide from EEF to support research are only made available by your support. So some housekeeping. Chat is disabled. Use the Q&A function to ask questions. If possible, refrain from asking personal health questions. And please feel free to use the show subtitles function for closed captioning. So I now have the honor of introducing Dr. Jose-Alain Sahel, Chairman of the Department of Ophthalmology at the University of Pittsburgh School of Medicine. He is the Ioneer Foundation Endowed Chair, and he is also exceptional class professor at the Sorbonne University in Paris. Dr. Sahel.
Well, thank you, Rita. Thanks for everyone for attending this webinar. Well, as you know, our department and University of Pittsburgh has a very specific focus on optic nerve due to the fact that we host the Fox Center funded by Reese Fox, and that is focusing specifically on conditions that affect the optic nerve and the ability to regrow and regenerate the optic nerve that is affected in many specific conditions like trauma, glaucoma, and tumors, and many others. This has been considered for a long time until very recently as an absolute limit for anything we could do to cure disease, because the optic nerve being part of the brain was considered as impossible to regenerate, and mostly preventive approaches were being taken, trying to protect the cells that form the optic nerve that originate in the retina and are connecting the eye to the brain. But because of the importance of reconnecting the eye to the brain and being able to keep this connection, the focus on research on that has been very important for at least the past 10 to 15 years. And the Fox Center has been a hub and a home for that. We're hosting a yearly meeting that invites world experts in the field and collaborators from many sister institutions like Harvard, Stanford, Paris, and many other places, but also more importantly, brainstorming on new areas of research. Over the past years, both at the clinical level with new recruits working on neuro-ophthalmology and specifically on optic nerve disorders, but also to the fact that thanks to the support of sponsors like Reese Fox, we were able to recruit scientists working on the optic nerve. We now have a very good team of several scientists working on these aspects, and their work in collaboration with others is already paving the way towards new innovative therapies. Interestingly, this is, and you'll hear Takaaki presenting that, and Dr. Kuwajima has been working on very promising drugs that could help in regenerating the optic nerve. One of the issues has been how to deliver these therapies to this region, which is behind the eye, which is not easily accessible. And we are very fortunate in Pittsburgh because Steve Badilak is a world leader in the field of material, extracellular matrix, and ways to deliver actually using these approaches drugs or any compound to, and forming also a scaffold for the regrowth of the optic nerve. So one of his lead students working specifically on this program is with us today and is going to present, Dr. Chang. So we'll have a joint presentation by the two of them. We have been working over the past year and a half on a very promising project that is supported by the Ioneer Foundation and donors like Reese Fox and also the Hillman Foundation to really help us to lead this program into a clinically relevant application. And as you will hear, it's early stage, but it's very promising. So I won't delay anymore the opportunity to hear from them. And I will hand over, I think, Taka, you're the first to speak, or is this Jordan? Taka? Yeah, okay. Jordan. Well, please take over from me.
Okay, Dr. Sahel, thank you very much for that introduction. Today, I'm going to talk to you about like he mentioned, an extracellular matrix-based treatment for optic nerve regeneration. My name is Jordan Chang. I've been working with Dr. Badalak for a couple of years now. I am a third-year medical student at the University of Pittsburgh. So like Dr. Sahel said, neuroinjury, especially optic nerve injury, has basically thought of as, almost impossible because of the poor regenerative capability of the central nervous system. And the retinal ganglion cells, which are the cells responsible for the optic nerve, their death is regulated in part by inflammation mediated by cells called microglia and astrocytes. So we have been studying the extracellular matrix for years now, and it was typically thought of as mostly structural components. It's one of the reasons why we look the way that we do, and we're not just blobs of cells. It's partly responsible for maintaining the shapes of organs and everything else that's in the body. But recently, or over the last 20 years or so, there's been more attention paid to the immunomodulatory aspects of the extracellular matrix, meaning that if we can scrape away all of the cells from organs and isolate just the extracellular matrix, that has immunomodulatory phenotypes, and the most important of which is that we've studied is macrophages and microglia, which are we're able to mitigate the inflammation from those cells and promote more of a constructive remodeling as opposed to chronic inflammation. Extracellular matrix has been used in over 10 million patients already in a variety of different clinical applications. Recently, we have identified a unique vesicle in the extracellular matrix that we have called matrix-bound nanovesicles. They are somewhere between about 100 and 300 nanometers in diameter, and they contain things like proteins and microRNAs and lipids on the surface that have immunomodulatory effects. And on the right side here, you can see images. This is a transmission electron microscopy image of these MBVs. And on the bottom there's a scanning electron microscope, and they're attached to the collagen. And we've found that there's ways to isolate these vesicles from the extracellular matrix. Importantly, the matrix-bound nanovesicles are able to recapitulate the effects of the whole ECM on macrophage phenotype, and that is moving away from inflammation and more towards regeneration or constructive remodeling. So some of the work that we've been working on here has to deal with the retinal ganglion cells. And this is a paper that was published pretty recently from our lab. And this shows that these MBVs are able to increase axon regeneration or neurite growth compared to controls. So on this right image here, you can see all of these nice projections. I hope you can see my cursor. See all these nice projections compared to just controls. And importantly, on the graph on the bottom here, these MBVs are not toxic to cells, so they're very healthy. We have used somewhere in the range of 10 to the 12th MBVs, and we have not been able to kill cells. So they're very, very good for cells. I know there's a lot going on this slide, but the important take-home message of this slide is that the MBVs are able to suppress pro-inflammatory glial signaling, and that's the astrocytes and the microglia. After these cells are already in their inflammatory state, if we give them MBVs, what ends up happening is they revert away from the inflammatory state and towards the more regenerative state. This is kind of a schematic of the in vivo model that is associated with this project. Essentially, what we did was we were able to attach a saline reservoir to rat eyes to damage the optic nerve. And the normal pressure of the rat eye is about 15 millimeters of mercury, and we were able to elevate that to 130 for about 60 minutes to really create damage to the optic nerve and the retinal ganglion cells. These MBVs were then delivered intravitreally at the time of the injury, 2 days after that, and then 7 days after the injury. And what we were able to see is that the MBVs are really able to decrease the intraocular pressure or IOP-induced retinal ganglion cell death. So on the left image on the left panel, we have the control healthy nerves. So just to the right of that, we have the IOP elevation. To the right of that was a simple control, and on the right image, we see the IOP elevation plus the MBV treatment. So really, you can see that after we give our MBVs, the retinal ganglion cells look much more like the healthy controls than they do the damage, suggesting that we are able to decrease the retinal ganglion cell death. And on the right side is just a quantification of that data. Another important part of the study is we're able to decrease the axon degeneration. So I'll go in the middle slide first. This is a cholera toxin, which just is going to label the entire nerve. The control on the top is just the healthy nerve. The IOP on the second frame in the middle image is what happens after the injury. The third is after the injury and a control treatment. And the last one is after the IOP injury and the series of treatments. You can see that it's really much closer to the control healthy one, suggesting that we are able to at least prevent axon degeneration, if not get some regeneration. The image on the left tells the similar story. That is just the labeling of the retina as opposed to the optic nerve. And the right side is, again, just a quantification of this data where there is a statistically significant difference between the treatment groups. This is, again, another histological staining image, and it tells a pretty similar story. GAP43 is a marker for neuronal regeneration. So I'm looking on the left image, on the left panel, the uninjured control does have some positive staining, but when we compare the MBV treated animals to the non-MBV treated animals, we do see markers of neuronal regeneration, suggesting that we are able to regenerate the optic nerve. And this is another staining image for glial fibrillary acidic protein. And what that is, it's a marker of inflammatory astrocytes. So here we see a lot of inflammatory astrocytes in the injured animals with the IOP and with the controls. But when we add the MBVs, we do not see astrocyte inflammation, suggesting that we are able to modulate the phenotype of those astrocytes. We were also able to conduct electroretinography, and just very briefly, the left image, the right eyes were the injured eyes, and the left eyes were uninjured. So you can see that after the injury, we lose ERG amplitude, and after we treat with the MBVs, we're able to regain a significant portion of that amplitude, suggesting that these cells are healthy and functioning again. So currently we're in stages of doing this in a large animal study with collaboration at Wake Forest. And what they did instead of this model that we did, it was an optic nerve crush. We're currently analyzing samples from this, but it's kind of a similar study design where we do the optic nerve crush. We inject our MBVs plus Dr. Kuwajima's statins, which he will tell you more about in a minute. And we will analyze it in a similar way with histological staining and electroretinography. And we're really excited about this study, and we hope to have results soon. We are analyzing the first 2 samples of this. And yeah, this is a really exciting project. I think we will take questions after both of us speak, but Dr. Kuwajima, if you are ready, you can take the screen from me.
Are you unmuted, Taka?
Yep. So thank you very much, Jordan. Thank you for Dr. Sahel for a great introduction. So my lab is focused on the optic nerve regeneration after optic nerve injury and also neuroprotection in glaucoma. So we just started 4 years ago And then we have a lot of exciting data here. So that's why we are very happy to share published and unpublished data today. So our research goal is to prevent RGC death and then regenerate the optic nerve and then reconnect the eye to the brain after ocular trauma. So as you see that after injury, you cannot see any axons anymore. So our mission is to identify effective and safe drugs and molecules to achieve the goal. So many scientists have been identifying different molecules that maintain RGCs and promote axon regeneration. So for example, cyclic AMP, PTM-TOR, SOX3, and STAT, and also other molecules, cytoskeleton signaling pathway, and also cytokine signaling pathways. And also other factors, transcriptional factors, KLF and CMYC, SOX11, and most recently, Yamanaka's transcription factors involved in this context, and also promoting axon regeneration. The most important thing is that, however, there is no effective drugs and surgeries to prevent optic nerve damage and neuronal death in the eye, but also to promote axon regeneration after ocular trauma. So we focus on the interactions between the injured axons and also factors in the environment. So as you see in this picture, in the normal mature central nervous system, axon is already connected to the target. And the non-neuronal cells, oligodendrocyte, astrocyte exist around the axons. However, after optic nerve injury or in the spinal cord injury, so axons start to degenerate. And the oligodendrocyte, myelin debris, and also reactive astrocyte, glial scar exist around degenerative axons. So to identify new molecules and new drugs, so we focus on the one of the inhibitory molecules for axon regeneration, MAG, myelin-associated glycoprotein here. So when I was in the Chris Henderson's lab at Columbia University as a postdoc, so we set up the axon outgrowth culture system here. So we plated neurons on the MAG expressing cells. And the axons actually inhibited by MAG compared to the control cells. And we use this culture system to screen for compounds that overcome inhibition by MAG among 58 chemical compounds. And we finally found that statins are the best candidates. So as you know, that statins are reduced cholesterol levels and the risk their cardiovascular diseases. However, nobody found that the statins could be involved in the axon regeneration. So there are 9 statin brands. Each statin has a different efficacy. So that's why we just test which statin brand is the strongest axon outgrowth using this culture system. And we found that servastatin is the strongest one, and the fluvastatin is the second strongest one. So to have the direct evidence that statins promote axon regeneration, so we set up the mouse optic nerve crush mouse experiments. And we performed the optic nerve crush and also intravitreal injection of PBS as a control or servastatin. And 2 weeks after injury, as you see, servastatin promote axon regeneration compared to the control like PBS. However, we still have several issues. For example, regenerative ability is still limited. And also RGC protection is weak. And servastatin is not available in the market anymore because strong side effects. So to overcome these issues, so we have a great privilege in Pittsburgh here. So we set up an exciting collaboration with Steve Bodylock and also Jordan. So his lab is focused on MBB, as Jordan mentioned that. But I just want to highlight here again. So MBB is a critical component of ECM extracellular matrix bioscaffolds. MBB changes immune responses and regulates neuronal survival and growth. One of the interesting findings here, MBB is a neuroprotective reagent in the mouse model of glaucoma. So several components of ECM are used in clinical therapies, and more than 60 FDA approved and used in clinics, and also more than 8 million patients treated with the ECM bioscaffolds. So based on this evidence that MBB could be approved by FDA in the future. So our current project is to investigate whether a new combination, fluvastatin, which is strong axonal growth effect, and also still available in the market, and the MBB together can enhance axonal regeneration and neuroprotection. So this is experimental design here using the mouse optic nerve crush surgery. So we use 8-week-old mice, and then we crush with the forceps for 5 seconds, and then 1 millimeter behind the optic disc. Just right after injury, we inject into the eye PBS as a control, MBB alone, fluvastatin alone, or MBB plus fluvastatin. And then 2 days after injury, also we inject again. And then 2 weeks later, we sacrifice the mice and analyze the RGC protection and axonal regeneration. So here is unpublished data. So we analyze the neuroprotection first. So we counted ganglion cell numbers stained with RBPMS, and the top of the these panels, is in the peripheral retina, and also this is central retina, and also VT, DT, DN, BN is a different regions. So in the injured retina treated with control like PBS, you appreciate the small number of surviving RGCs here. However, the injured retina treated with MBB fluvastatin shows increased number of the surviving RGCs in the all regions, in the peripheral retina, and also even in the central retina. So this is quantification data of the RGC survival rate. So for example, their statin has a minimal neuroprotective effect, like here. However, MBB has a protective effect. But surprisingly, MBB fluvastatin combinations shows more robust neuroprotective effect. Next, so we analyzed the axonal regeneration 2 weeks after injury. So as we expected, fluvastatin promote axonal regeneration. And the MBB, as Jordan mentioned, that promote axonal regeneration. But unexpectedly, MBB and the fluvastatin combination therapies shows more robust axonal regeneration compared to the single treatment based on the number of regenerated axons here. So based on this exciting unpublished data, so we found that MBB statin promote RGC protection and optic nerve regeneration. So currently we have been using the RNA-seq techniques and then to identify molecules and then molecular mechanisms right now. So hopefully we're gonna provide exciting and new molecular mechanisms by MBB and fluvastatin. So finally, I really thank my labbers, especially the Greg Campbell for this project, and the great collaborators, Steve Badilak and Jordan, and also Jeff Brooks. And then finally, we really thank Dr. Sahel and Mr. and Mrs. Fox and the other founders for generous support. Thank you very much. We are happy to take any questions.
Wonderful presentation. Thank you so much. We do have quite a few questions, but my first one is, let's talk about the collaborative spirit of the Louis J. Fox Center for Vision Restoration. It collaborates with the McGowan Institute, but also other universities, and the meeting that's held every year to attract different researchers to talk about optic nerve regeneration, gets people from California, Florida, whatever. So if we could talk a little bit about the collaborative spirit of this center, that would be a great place to start.
Sure, maybe I can start. So the The center was started many years ago, more than a decade ago, and it was really prompted by the will from Louis Fox to promote this type of research, especially in the field of regenerative medicine with a specific focus on optic nerve regeneration, but also beyond that. As I alluded to that initially, there is a yearly meeting that has become virtual recently, but was an in-person meeting that brings together scientists from all over the country, from leading institutions, but also from all over the world, and a small group of people, not a large number. Like you need, if you are more than 20 people discussing, it becomes totally ineffective. But really, the leaders and exchanging ideas, and given the many challenges of optic nerve regeneration, this needs a lot of thinking and a lot of innovative out-of-the-box initiatives. And this is what has happened, and it has led to many collaborations, especially we have very good partnership with Stanford, Harvard, Paris, and other locations. But importantly, because of the McGowan Institute and because of the abundant number of top scientists in Pittsburgh, there is now a real strong emphasis on the collaboration with a group of Steve Badilak, and also within the Department of Ophthalmology, Chang has been recruited from Stanford. He's also working on optic nerve regeneration and works daily in collaboration with Takaaki. We have also Issam Aldiri, who is working on genomic aspects of regeneration, and many other scientists in the department. So the science is beyond borders, and having all these talents together. And today, the illustration of the ability to identify new drugs, but coping with toxicity, release, and delivery using the new technology developed by the group of Steve Badilak is a good example of what the Fox Center can promote. And actually, I have to say that the initial meeting to trigger this collaboration was something that resulted from a Louis Fox initiative telling us you have to sit down together and start the project. I can remember exactly when and how it happened. So it's an example of that. Yeah.
Thank you. So, we talked about a little bit about your current research, Taka, that you're working on with Dr. Badilak, and it involves Wake Forest University. Could you talk a little bit about your work with Wake Forest? Just the collaborative efforts between the Lewis J. Fox Center and Wake Forest.
Well, it's because you need a large animal model, and because you need to validate that what is working in small animals, given the size of the eye and for many reasons. And the Wake Forest, they have developed this model, which is, and so it's better not to duplicate that. And this is why we have raised this collaboration. Maybe Taka, you may want to explain a bit more on that.
Yeah, I think Dr. Sahel explained a lot, so that's why I don't need to add anymore. But I think my expertise is using the mouse, and also we have the knowledge about their optic nerve in the mouse. We know a lot of their like cells or those kind of stuff, but we have no expertise in the large animals like a pig. So that's why Wake Forest, they have a great team to do the optic nerve crush in the pig, and then they just do surgery, because the optic nerve and the eyeball is much bigger than mouse optic nerve and the eye. And also that's actually you need special techniques or like procedures. So that's why we are not working on here. So that's why we need a collaboration like this. So that's why we are very happy to this collaboration.
Thank you. So we have some clinical questions. As a glaucoma patient with extensive vision loss, how close am I to being able to be treated with this type of therapy?
Maybe I can start with that. So it really depends on how many ganglion cells, the cells that are forming the optic nerve are still present in the retina. If we are talking about a fully terminal stage of glaucoma where all the ganglion cells have disappeared, it's probably too late. And this is where the work of Kunche, for example, in the department, trying to use transplanted cells to really regenerate would apply. And then there would be a synergy with the program that we are discussing today, because then you would transplant the cells and help them to regrow axon. But this is something which is like a bit already a lot of work in the making, in progress, but not yet there. But you still have the ganglion cells with cell bodies, which means the cells are still alive, at least enough of them are still present in the retina. then this therapy could be contemplated for this patient. Currently, the approach to glaucoma is to prevent optic nerve damage, and we cannot emphasize enough how important it is to be treated on a regular basis, monitor the intraocular pressure, make sure that the levels are constantly being controlled, because this is the only way currently to protect the optic nerve. The intraocular pressure is the main risk factor for glaucoma. But many patients are not treated very early. Many of them, the therapy is not always applied fully, and some of them escape therapy, and there is damage to the optic nerve. And this is where this type of therapy is so promising. And I was looking at the graph that Taka was showing. I'm amazed actually at how powerful this is. I was hopeful it would be like additive, but it's more than that. It's synergistic. It looks like we are reaching a level that I couldn't even think of. So I don't want to be over enthusiastic, but this is certainly something that could be an approach for people who still have some ganglion cells that could be regenerated. Don't know if you want two of you to comment on that, Jordan or Taka.
Okay.
I don't think I have anything to add. That was well said.
Very good.
Okay, this is an interesting one. Are there any treatments for optic nerve hypoplasia?
Well, hypoplasia is a congenital situation, which means that the optic nerve has not developed. So, I mean, certainly not regenerating because the cells are not even there. But this is the type of approach that the day we are able to transplant cells and they are able to regrow could be contemplated, but we are not yet there at all. So it's something which it's a congenital condition, and some of them are very mild. Some hypoplasias are not very important. So then I think maybe this could be contemplated, but in some cases it's a very, very severe loss of, actually not even loss, but not even formation of the optic nerve. And probably this is beyond the scope of protection or regeneration. It's really a new field with transplantation, but nothing is impossible.
That's wonderful, that nothing's impossible. So can the MBV treatment be used for optic nerve damage from an aneurysm in the carotid artery?
Well, I mean, yeah, please, Jordan.
It's something that I'm not too sure about, but if, the model that we are using is kind of a model of ischemia. So if you had a loss of blood flow to the retina, it's possible that that could be pretty close to the model that we are doing. So the MBVs could potentially be used as a treatment. But in terms of, that specific condition, I think it would require a lot more research about, that specific model before we can know anything more.
That's great, thank you. Now here's one for all of us who take statins. If statins have been shown to help generate the optic nerve, is there any evidence to suggest that prior statin use could also help to prevent further optic nerve damage?
Taka?
Yep. So that's really a good question, and also it's really important question. Actually, so people usually take their statins by mouth orally. So right now, 8 statins are available, and then not all of them actually cross a BVV. That means that do not going to the brain, do not going to the eye or spinal cord. So that's why it's just blocked. And so that's why even though, so for example, 2 of 8 statins are cross BVV. So that means that could be useful for treatment in the brain or optic nerve, but we have to choose really carefully. But also nobody knows about their treatment, the effects in their in the human patients yet. So that's I cannot say that it's effective or not. But one thing is that in glaucoma patients, actually the statin users delay the disease progression without any controls of the IOP changes. But still, these data are controversial. So I'm not saying that this is correct or not. But we still get more knowledge and information. Hopefully, we're going to provide something more interesting, more useful data in the future.
I'll Heather, I think your air conditioning is running, so it's making a lot of background noise. So I'll finish with some of these questions. Hi, folks. Lawton Snyder with the Eye and Ear Foundation. And thank you, guys. This has been a very interesting, and I see we have so many questions. I will tell the audience some of the questions we may get back to you via email, because they would be very specific to maybe your condition, and it may be better for us to answer you that way in terms of this presentation. But here's one that's kind of focused on someone who seems to know a little bit more about the work you're doing. Is the dose of MBV intravitreal injection, is the MBV number based on its cargo content based, like doing a simple BCA to know the protein concentration? That's obviously something for one of the two scientists here, probably Jordan.
Yeah, that's a really good question. So we go by particle count. We got a machine that will actually count, almost each individual particle. So we will go based off of the number that we are using right now is 1 × 10⁻⁻⁻ MBVs, and they're very small. So we can get that into about 1 microliter. So yeah, we used to do it based on protein content, but then we were able to, get a machine to actually count the individual particles. So yeah.
Thank you. I see a lot of questions wanting to know if this is available for humans yet. And of course, we have to tell you, it's not available for humans just yet. Obviously, that's everything we're working towards. And the faster we can do the trials through the larger animal models to some potential work towards clinical trials, obviously, we'll hope to have that happen. But but we certainly want you to know that we're working towards this, and you can understand that the process is not always predictable. But there's a question here, and maybe I don't know if this can be extrapolated. Can we determine whether or not long-term damage from optic nerve? So if you had not just a recent injury or recent vision loss, but something that happened years ago, does this type of potential therapy that we're talking about have potential hope for people in that condition?
Well, I think the answer is similar to what I said a moment ago for glaucoma. It depends on the stage of a disease. You can have a long-standing disease, but if you have remaining ganglion cells, there is still a potential that this regrowth of the axons could be attempted and contemplated. If the cells are totally degenerated, we are talking about end-stage disease, probably beyond that. This is why we have all this axis of research on transplantation to replace the cells, but it's a different axis. It's synergistic with the work that is being done by Takaaki and Jordan with Steve, because if you transplant the cells, you need them to regrow the axon. So it's not that it's one or the other, could be the two of them, but it's like a two-step process, transplanting and regenerating.
Okay. And I think the rest of these is, again, we have a lot of the same questions. I can understand it. Everybody's hoping we have this soon. We do too. We hope that we have it very soon. And another one here, maybe here for you, Jordan, is cargo content related, so that I can identify the regenerative molecule. Are you looking to see whether this is an miRNA protein in the MVP?
We are looking into that. There have been a number of microRNAs that have been identified in the MBV. There's also a lot of proteins on the surface, and both in the lumen. And we've also seen, we've also analyzing lipid content to see if there's something actually just in the cell membrane that could be responsible. The short answer is we don't know right now, but we are looking into it. And as far as that last question with the kind of delayed treatment, I know we're currently doing 2 studies here. One is where we are doing the similar injury, but we are waiting 3 days after treatment. I know it's not years, but it'll give us some sort of an idea if we are able to heal past injuries. And then the other study that we're doing here is a glaucoma model where we're letting the damage kind of accumulate for a month or two before we intervene. So that hopefully will give us data that will answer your question, but unfortunately we don't. It's in the early stages, and we hope to have it soon, but we don't have it right now.
Yeah. And maybe this helps some of those folks asking that question in terms of the process. When a treatment as we're talking about, may become available, if you're going to be using an MB, B or a combination therapy like we're talking about, is it going to be a very invasive process? So I guess in a sense, if your damage was severe enough, would you, and obviously based on the clinical trial entry criteria, would it be an option for people to be considered for something like this if they are, based on the invasiveness of the procedure?
But my understanding is that intravitreal injections are fairly common in clinic. They use it for diabetic retinopathy to give treatment that way. So I don't know the specifics. Dr. Sahel would probably know, but I understand that it's maybe a little bit of minor discomfort the next day. It's not really too invasive of a treatment.
I agree. I mean, if it is intravitreal injection, it's not an issue at all.
That maybe gives some people some ideas. It may be something that could be tried for people with, as we get to the point where it can become something of the clinical trial. All right, I think again, the rest of these, we've had a lot of the similar questions. People want to know if, and I'll maybe refer to Dr. Sahel for the NION question. And of course, that's another type of optic neuropathy. And we're talking about the same thing. If it's an optic neuropathy, this would also be something that we'd be hoping could be used for that purpose. Am I right, Dr. Sahel?
Yeah. It's an ischemic condition. So as Jordan said, the model is a model of ischemia too. It really depends on how severe the damage is to the cells, but potentially this could apply to that condition too.
Okay. And we only have one more here that we haven't really answered otherwise. And I don't know if it's, if the audience would also want to hear, but I don't know anything about these herbals. There's a question about Chinese medicine. Kuwajima and Yugui. Have you heard of any success using these herbals? I guess it's from a study that was also published. I don't know if any of you have had any. It may not be something we're looking into here or have any knowledge of, I'm assuming.
Well, it depends on wherever it was published and what journal and the quality of the data, but I'm not aware of a study that made a mark in the field that it's working.
Using any of these herbals. Okay. Yeah, obviously, stay tuned for what we're working on in the Fox Center related to research that's going on. We will continue to give these updates as we have through our webinar series, as well as through our publications, which you can, if you sign up for the webinar, you should also have an opportunity to receive some of our information via online newsletters and so forth. So if you, and certainly feel free to email us through Craig Smith, who you receive the information from, if you have any additional questions. But thank you both, and thank you, Dr. Sahel. So this has been a very good update. There's a lot of promise is where we are right now. We've reached some stages that I think everybody's very excited about. And I love to hear Dr. Sahel get excited. He sees a lot of the great research around the world. And when you see that this is something that has raised his eyebrow in terms of the opportunity and the potential, I think that also gives us all a little bit of hope that this next stage can be very important for what we want to learn, what we want to see in a larger animal, and then maybe, from there, hopefully a clinical trial in the future. So we'll all hope that science rolls out to be the lean in our favor and give us something that will benefit patients with optic nerve damage. That's the goal. Thank you all, and have a wonderful rest of your afternoon.